Hello everyone. Today we'll be solving Cambridge International AS and A-Level Biology Paper 4 A-Level Structured Question May-June 2021 Paper 4-1. Question number 1A. Photosynthesis is a complex process involving the transfer of light energy into chemical energy. Describe the role of photosynthetic pigments. Photosynthetic pigments absorb light. Accessory pigment passes energy to primary pigment, and the pigment forms a light harvesting cluster called photosystem. The light energy is used to excite electrons for cyclic and non-cyclic photophosphorylation. Part 2. Name the precise location. Name the precise location in a chloroplast of photosynthetic pigments. Photosynthetic pigment can be found in thylakoid membrane. Part 3. Name the practical technique to separate photosynthetic pigments. So to separate photosynthetic pigments, we can use chromatography. The rate of photosynthesis is affected by many environmental factors. A student carried out an experiment to investigate the relationship between the concentration of carbon dioxide available to an aquatic plant, Cabomba caroliniana, and its rate of photosynthesis. Figure 1.1 shows the experimental setup for this investigation. We can see the LED lamp is placed at a fixed distance, and in a sealed container there is a 15 centimeter length of caroliniana, and there is sodium hydrogen carbonate solution to provide carbon dioxide in this aquatic environment. Sodium hydrogen carbonate solution was used as a source of carbon dioxide. The concentration of carbon dioxide was varied using 6 different concentrations of sodium hydrogen carbonate solution. All C. caroliniana plants were kept in dark before the light was switched on at the start of the experiment. 5 replicates were carried out at each concentration. The rate of photosynthesis was obtained by calculating the percentage change in the dissolved oxygen concentration in the solution over 5 minutes. Figure 1.2 shows the results of the investigation. So we can see as the concentration of sodium hydrogen carbonate solution increases, we can see the rate of photosynthesis increases because percentage change in dissolved oxygen increases. However, then it plateaus. With reference to figure 1.2, explain the pattern of results obtained between 0.25 mol per dm cube and 1.25 mol per dm cube sodium hydrogen carbonate solution. So we want to know the pattern ranging from here to here. Okay. So from 0.25, if we notice from 0.25 mol per dm cube sodium hydrogen carbonate, once we increase to 1 mol per dm cube, once we go up until 1 mol per dm cube, concentration of CO2 is the limiting factor, because as we can see, if we increase the concentration of CO2, the rate of photosynthesis increases. Greater than 1 mol per dm cube light intensity or temperature could be the limiting factor, because at that point sodium hydrogen carbonate or carbon dioxide is no longer the limiting factor. Before 1 mol per dm cube, CO2 is the limiting factor because it is the raw material for photosynthesis, which is needed for light independent stage. Part 2. The percentage change in dissolved oxygen for C. caroliniana at 0.00 mol per dm cube of sodium hydrogen carbonate solution is negative. Suggest reasons for this negative value. So basically at 0.00 mol per dm cube sodium hydrogen carbonate, there is no carbon dioxide. So there is no photosynthesis. Alright, aerobic respiration only occurs, which uses up the oxygen. Part 3. To minimize temperature changes, the student decided to use an LED lamp as a light source. LED lamps release very little heat energy. Explain the importance of minimizing temperature changes in this experiment. So basically, temperature is a control variable in this experiment. To ensure a fair test, CO2 should be the only variable, all right, and the rest of it must be kept constant. Low temperature would decrease the rate of photosynthesis and oxygen production would decrease. High temperature would increase kinetic energy of reactant and increase O2 production. Too high temperature may cause photorespiration. Lower temperature is a limiting factor, so can limit the rate of photosynthesis as well. Guys, as you notice, these particular questions have more than the number of points that actually needed to score that particular mark. So because of that, if you want to avoid 1 or 2 points from here which doesn't feel relevant to the particular answer, then you may just simply avoid it and only do the minimum number of points for scoring the mark. However, writing extra point allows you to get full marks in the exam, because even if you lose 1 point, you will get mark in another one. Question number 2a. All organisms respire to release energy from energy-rich molecules such as glucose. Anabolic reaction use energy. State 2 examples of anabolic reactions occurring in living organism. In living organism, there is always DNA replication that can take place if a particular cell wants to divide. All right, then to make protein, because all the time protein synthesis takes place. All right, also if cell membrane synthesis takes place, so to make phospholipid. Part 2. Describe and explain the use of ATP in glycolysis. So basically, ATP phosphorylates glucose into hexose phosphate. ATP then phosphorylates hexose phosphate into fructose bisphosphate, so that glucose cannot leave the cell. ATP provides activation energy to break down the glucose. Some factory workers in the early 20th century were exposed to chemical X and experienced serious side effects. Some of the effects of exposure to chemical X are decreased production of ATP, increased lipid metabolism with weight loss, increased production of pyruvate and lactate, excess heat energy released causing an increase in body temperature which can be fatal. Chemical X increases the permeability of the inner mitochondrial membrane to protons, hydrogen ions, causing some protons to leak out into the matrix. Explain why people exposed to chemical X shows decreased production of ATP, increased lipid metabolism. Okay. So guys, for a basic understanding of this particular question, we must understand the structure of a mitochondria. So in a mitochondria, there is a outer membrane and then there is an inner membrane. The inner membrane is folded to form cristae, which basically allows it to do oxidative phosphorylation in the inner membrane, as the electron transport chain and other proteins needed for it are housed here. So for the electron transport chain, there will be proteins which will then lead to an ATP synthase as well. Typically what happens is that whenever a NADH comes in, it releases hydrogen ion, and those hydrogen ion, all right, is pumped. It is pumped into this intermembrane space, and when those hydrogen ion comes down the concentration gradient through the matrix, all right, so basically that energy is used for combining ADP and PI into ATP. So basically, all right, if the H plus ions are able to come from the intermembrane space into the matrix directly, this is going to release heat energy. Also, as the membrane is more leaky, as the inner membrane is more leaky, what happens is that enough concentration of H plus ion cannot build up. So ATP is also not synthesized by the ATP synthase. So that's basically the explanation. So the answer will be ATP production decreases due to less steep proton gradient. So fewer protons go through ATP synthase, lipid metabolism increases and uses body fat stores as less ATP is made from glucose. Part 2. Suggest and explain why chemical X causes increased production of pyruvate and lactate. So basically chemical X causes faster rate of glycolysis. Oxygen is used faster, so increased anaerobic respiration. Part 3. Suggest why excess heat energy is released. So basically energy is not used to join ADP and PI, so it is released as heat energy. Question number 3. In 2014, scientists published a phylogenetic evolutionary tree diagram showing how and when different groups of insects developed from ancestral groups. To construct the diagram, the scientists compared nucleotide and amino acid sequence data for 1478 genes from each of 103 different species. The 103 different species represented all 32 orders of the class Insecta. The data were used to arrange the species in related groups according to the degree of molecular similarity and to track how sequences changed over time. Outline 3 ways in which the member of a species are similar. So basically, a member of a species are similar morphologically, physiologically, and biochemically. It can also be genetically they are similar. The member of the same species have same niche, meaning that they have the same role within an ecosystem. They have a similar behavior. They interact with each other in a similar manner. They can interbreed to produce fertile offspring. The members of species are reproductively isolated from other species. The members share evolutionary lineage or common ancestor. Guys, I want to still give you a reminder. You do not have to write so many points to get the full marks. Each change in color indicates a particular marking point. So you can just write simply 3 to 4 points to get this full marks. Organisms are classified in a hierarchical system of taxonomic units. Name the domain and kingdom to which insects belong to. So basically insect belongs to eukaryotes because their cells have a nucleus. Alright, and kingdom is Animalia because they're animals. Explain why phylogenetic tree diagram is useful for classifying insect species. So basically classification depends on evolutionary relationship. Alright, organisms that are closely related, they share common ancestor, so should be classified together. And phylogenetic tree diagram help identify larger taxonomic group. Part B. For this study, new software for processing large quantities of data was developed. Calculate the total number of sequences that were compared, considering both the nucleotide and amino acid sequence for each gene. So we had a total of 1478 genes. So there is 1478 genes. All right. And then there were 103 different insect species. So multiplied by 103 different insect species. All right. And then there were nucleotide and amino acid sequence. So multiplied by 2. So there is a total of 304468 combinations. 304468. Explain why this study needs to make use of bioinformatics. So basically this study needs to make use of bioinformatic techniques to store, analyze large quantity of data, to compare many sequences, to calculate percentage similarity and differences within the sequences. In a fast and accurate and efficient manner, we need bioinformatics. Question number 4. Genetic engineering involves the manipulation of naturally occurring enzymes and processes. List the enzymes used in genetic engineering, genetic modification, and outline their roles in natural processes. So we know genetic engineering uses restriction enzyme. So the restriction enzyme is used for cutting the DNA into fragments. The restriction enzyme is produced by bacteria, and basically it's actually a evolutionary it's like an evolutionary thing for bacteria, because they want to fight, they want to defend themselves against bacteriophages, which is why they produce restriction enzyme. DNA ligase, alright, joins DNA together to form phosphodiester bond. DNA ligase is needed during DNA replication to join the Okazaki fragments together. Reverse transcriptase forms cDNA from mRNA template. Reverse transcriptase is present in retroviruses. And Taq polymerase, it replicates DNA at a very high temperature, and Taq polymerase is produced by Thermus aquaticus bacteria found in hot springs. Part B. Genetically modified organisms include crop plants. Genetically modified plants have been grown in North America in increasing quantities since 1996. They are now grown in many areas of the world and are eaten by millions of people and farm animals. Table 4.1 compares the area of land used to grow GM plants on 6 continents in 2013. Africa. Alright, area of land used to grow GM crop plants in millions of hectares, 3.5. Edible GM crop plants grown maize, soybean. So we can see that in Asia there is 19.1, 0.6, Europe 0.2, 81.1, 70.9. Alright, so let's carry on. Suggests how North America and South America benefit from growing large scales of GM crop plant rather than non-GM crop plants. So basically here, it doesn't matter with the country, all right. It's basically just we have to give the advantages of genetically modified crops. GM crops produces more yield at harvest. All right, GM crops produces better quality of food because they are they don't require much use of insecticide or pesticide. GM crops requires less land to grow more amount of food, so areas can be used for conservation of animals and other things. GM crops have high yield, which means less fossil fuel is used per ton of crops produced. Only a small area of land is used to grow GM crops in Europe. This is mainly because most of Europe uses precautionary principle. The precautionary principle prevents GM crop plants being grown if there is a possible risk of harm to human health or to the environment, even if there is no proof of harm. Many European citizens have concerns about the safety of eating GM food. But Europe imports large quantity of GM maize and GM soybean to feed farm animals. Use this information in table 4.1, deduce 2 arguments in favor of growing more GM plants in Europe. So basically, people in other continents, example Asia or North America, they already eat GM crops. All right, so technically there is not much of a health benefit of not eating non-GM crops. All right, GM crops is already in European Union food chain because the animals are feeding on them. And there will be a great economic benefit and increase in self-reliance if GM animal food import is reduced. Guys, as you can see, this particular question's answer contains 3 marking points. So, if you want to get only 2 marks and you want to write very little, then just write down the first 2 points. The third point will not be awarded anyway because these 2 points are included together, so will be rewarded as 1 point. Part 3. Explain why the data in table 4.1 are not enough to calculate the extent to which different continents have replaced traditional crop plants with GM versions. The problem with this previous table in this particular page is because this particular land area that we can see in millions of hectares, we do not understand whether it is, what percentage of the total area of the land that they are covering in that particular country. because different countries have different sizes and thereby different amount of agricultural land. So we need to know total crop area so we can work out the percentage fraction of GM to non-GM crops. Question number 5. A nerve consists of bundles of different types of neurons. Figure 5.1 shows the mean transmission speed of impulses for three types of sensory neuron. C, A with delta, and A with beta. So we can see mean axon diameter and the mean transmission speed of impulses. As the axon diameter increases, the transmission speed of impulse increases. Impulse from the pain receptors in the skin sent along sensory neuron C and A delta. Sensory neuron C and A delta synapse in the spinal cord with relay neurons known as projection neurons. Projection neurons send impulses to the part of the brain that perceives pain. Impulses from touch receptors in the skin pass along sensory neuron A beta, which can also synapse with the projection neuron in the spinal cord. Sensory neurons A delta and A beta are myelinated. Sensory neurons C's are unmyelinated. Describe the results shown in figure 5.1 and suggest explanation for these results. So we can see, all right. So in 5.1 we can see as the mean axon diameter increases, mean transmission speed of impulse increases. All right. A beta, all right, has 9 micrometer in diameter and a 55 meter per second speed, whereas C has 1 micrometer diameter and has a 2 meter per second speed. All right, and A beta has wider axon diameter, which increases speed of transmission of impulse. A beta has larger membrane surface area, so more ion movement can occur. Myelin in A delta and A beta increases speed of transmission for both of them. Solitary conduction in myelinated neuron increases transmission speed. Part 2. Gently rubbing a damaged area of skin can reduce the perception of pain. Suggest an explanation for this. Impulses for touch are A beta, which travels faster. So A beta neurons inhibit relay neurons in spinal cord. Part B. Back pain in humans can be reduced by using transcutaneous electrical nerve stimulation or TENS machine. A TENS machine uses electrical impulses to stimulate nerve endings near the site of the pain. It is thought that TENS triggers the release of natural painkillers called endorphins at synapses. Figure 5.2 shows synapses in pain pathway. So we can see impulses from area of muscle, painful back muscles are coming through here, and impulses from area stimulated by TENS machines are coming through here. These vesicles containing endorphins, and these vesicles are containing acetylcholine. All right, so pain center in the brain and postsynaptic neuron will respond to it. So basically, if TENS machine is working, then the TENS machine will release endorphins, which will then bind with the impulses from area of painful back muscles, and as a result, this particular acetylcholine will not be released, and thereby the pain center in the brain will not be activated. So suggest and explain how endorphins may act to reduce pain. So the answer will be like this. Endorphins binds to endorphin receptors and stops calcium ion from entering the presynaptic knob. As a result, fewer vesicles containing acetylcholine move towards and fuse with presynaptic membrane. As a result, less acetylcholine is released, so less acetylcholine binds to postsynaptic receptors. As a result, no depolarization of postsynaptic neuron and fewer action potential to pain centers occurs. Suggest one disadvantage of using pharmaceutical drugs for reducing pain compared to using TENS. So basically, using pharmaceutical drugs can cause drug dependency and side effects. So guys, that's all for up until question number 5. The rest, question number 6 to question number 10 will be solved in the next video. Please subscribe to our channel and press the bell icon so that you can get notification. If you like the video, then recommend it to your friends and let them get benefited from the video as well. And these question answers are by far the best points covering answers that there is. So if you memorize the answers and use it in a similar question like the one represented in this, you will definitely get your full mark. Thank you. See you in the next video. Bye bye.